So, Mesolithic 1 period, the blue-eyed female from Fertile Crescent AH1, Lorestan Province, Iran, a match 0.24% DNA. So, about the sample, AH1 is the scientific designation for the ancient genome of a prehistoric individual excavated from the archaeological site of Tepe Abdul Hossein in the Lorestan province of Iran, situated within the eastern mountain rim of the Fertile Crescent. Discovered in the central Zagros Mountains, this individual represents one of the world's earliest transitional communities, mapping the critical shift from Mesolithic hunter-gatherer to early Neolithic farming. The archaeogenic identity of AH1, sex female, chronological era datable to approximately 8,250 BCE to 7,700 BCE, the Aceramic pre-pottery Neolithic period. Mitochondrial DNA belongs to the R2E haplogroup. The blue-eyed context. While European hunter-gatherers are famous for carrying early genetic markers for blue eyes, paleogenic mapping across the broader Zagros and West Asian regions indicates that alleles linked to lighter eye and skin pigmentation emerged in mosaic pockets across the Near East. The broader Iranian Neolithic lineage contributed significantly to migrations that carried these diverse physiological traits westward into the Levant and eastward into South Asia. The geographic and cultural landscape, Lorestan Province and the Zagros. Tepe Abdul Hossein is situated on the slopes of the central Zagros Mountains. This rugged terrain served as a primary ecological hub for the independent development of agriculture and livestock management. The Eastern Fertile Crescent, the region was a crucial, separate cradle of civilization. Rather than agriculture spreading from a single point in the West Levant, communities here independently began domesticating local resources. Excavation layers associated with AH1 showcase a transitional economy. This includes early cultivation of wild barley and lentils, alongside some of the oldest genetically documented herded goats in human history. The global evolutionary significance. A landmark 2016 study published in Science used AH1 and neighbouring genomes to reshape our understanding of human migration. Distinct from European ancestry, AH1 belonged to a distinct population that separated from Western Anatolia groups between 46,000 and 77,000 years ago. They are not ancestral to early European farmers. Ancestors of South Asians, the genetic lineage of AH1 shows direct ancestral links to modern-day populations in Pakistan, Afghanistan, and especially modern Iranian Zoroastrians. Zoroastrians. Genetic continuity. The rugged landscape of the Iranian plateau functions as an isolation barrier. This allows the deep-rooted genetic legacy of early Zagros populations to be strongly preserved in modern regional populations. The burial method. AH1 was buried in a modest, highly localized fashion, typical of the transition from the Mesolithic to the early Aceramic pre-pottery Neolithic period in the Zagros Mountains. Like many individuals of her era, she was buried directly within the boundaries of the prehistoric settlement. During this period, the dead were routinely placed beneath the floors of homes, in open courtyards, or inside mud brick structures. This reflects a deep cultural desire to keep ancestors integrated within a physical framework of daily living spaces. She was buried in a tightly flexed or fetal posture. This was the standard mortuary practice for early Zagros populations, where the knees were drawn tightly toward the chest before placement into a small, simple pit. There was no monumental tomb or rigid alignment. Her resting place was an ordinary earthen pit dug into the occupational layers of the mound. Because AHI lived in a pre-pottery Neolithic culture, she was not buried with ceramic vessel metals or flashy valuables. Instead, the artifacts recovered from her immediate occupational layer and similar burials at Tepe Abdul Hussein reflect a simple, resource focused economy. Flaked and ground stone tools, simple chipped stone blades, scrapers, and ground stone implements were found. These tools were designed for day to day survival tasks such as harvesting wild greens and scraping hides. Grinding stones, mortars, and pestles used to process gathered wild plants, barley, and lentils were common fixtures of the living floors surrounding these early burials. Animal bones, contextual layers containing the remains of wild game and early managed fauna. These bones provide the critical evidence showing that her community was actively pioneering the domestication of local goats and sheep. So, how do I connect in DNA with this sample? The maternal lineage R2, carried by the ancient Iranian female AH1 and J1C3, are related as distinct branches growing from the exact same genetic trunk. They connect deep in prehistoric time through a shared maternal ancestor known as macrohaplogroup R. The common ancestor, macrohaplogroup R, time period approximately 56,000 BCE. Every single person carrying an R2 lineage or a J derived lineage shares a direct maternal line back to a single woman who lived in Eurasia over 50,000 years ago. Her daughters founded different paths. One path eventually led to AH1's lineage, while a parallel path evolved into the GT branch. The branching point R2 GT, time period 54,000 BC approximately. Within the R family tree, a specific transitional node called R2 GT formed. The fork in the road at this point, the family tree split cleanly into two directions. Path A, AH1's line, mutated directly into R2, which later formed the rare R2E subclade found in the ancient Zagros Mountains. Path B, the J1C's line, C3's line, mutated into the JT node, which acts as the direct parent to both haplogroup J and haplogroup T. And this lady wasn't found alone. The AH1 skeleton was excavated at the Tepe Abdul Hossein archaeological site, where researchers uncovered a small communion early farming settlement containing the remains of at least 12 Neolithic individuals. Her burial and the surrounding site reveal several fascinating details about how her community lived and handled their dead. Like many early Fertile Crescent cultures, this community buried the dead directly under the plasters of the homes, mud brick homes, or within abandoned rooms. Bioarchaeological analysis of the site shows a mix of adult men, adult women, sub-adults, and infants. Notably, one adult female at the site was found buried alongside a fetus, accompanied by a large stone bowl placed near her head. Several of the individuals found alongside AH1 showed clear evidence of intentional head-shaping cranial modification. This was achieved by wrapping bands around infants' heads to permanently elongate the skull as a marker of tribal or social identity. Interestingly, AH1's skull did not show this modification. Shared genetics. The landmark 2016 genetic study actually attempted to sequence multiple individuals from Tepe Abdul Hossein, while AH1 yielded the highest quality fully sequenceable DNA. Other skeletons from the exact same site confirmed they all belonged to the same unique isolated population of early mountain farmers. So the exact cause of death is unknown. Because she lived over 10,000 years ago during the early Neolithic period, her soft tissues are entirely gone and her skeletal remains do not show any definitive markers of fatal trauma. Bioarchaeologists study The population at Tepe Abdul Hussein can, however, narrow down her death to the standard realities of early agricultural life in the Zagros Mountains. Her bones showed no evidence of perimortal trauma, meaning there are no unhealed fractures, cut marks, or from sharp tools, or impact injuries that would suggest a violent death or warfare. The early communities of the eastern Fertile Crescent at this time were relatively small and isolated, and widespread tribal warfare was not yet a prominent feature of daily life. While agriculture brought stable food sources, the transition from foraging to farming drastically reduced human life expectancy. Skeletons from her community display physical markers of severe physiological stress. Infectious diseases settling in permanent mud brick villages created sanitation issues. Living in close proximity to domestic animals like early goats introduced zoonotic diseases and parasites to humans for the first time. Nutritional stress and anemia. Genetic and bone analysis of early Zagros farmers show high rates of nutritional deficiencies, relying heavily on early cultivated grains rather than a diverse wild diet led to widespread iron deficiency, anemia, which weakened the immune system. The high female mortality rates. Demographic studies of early Neolithic populations show a sharp drop in female life expectancy compared to the preceding hunter-gatherer eras. Women frequently died in their late teens, 20s, or early 30s. The spike in mortality is heavily linked to the complications of childbearing and the metabolic stress of frequent pregnancies in a nutritionally compromised environment. Given that another female at the exact site she was found buried, directly alongside a fetus, complications arising from pregnancy or childbirth remain a high plausible reason for her death. So AH1 was genetically connected to AH2 and AH4. They belonged to the exact same homogeneous ancestral population known to geneticists as Iran N, the Iranian Neolithic farmers. They lived at the Tepe Abdul Hussein settlement around the same time, 7,900 to 8,200 BCE, and shared almost identical global ancestry. However, analysing their specific maternal and paternal lines reveals their exact lineage relationships. So, the maternal connection. AH1 and AH2 are a direct match. Genetic tracking revealed that AH1, the female, and AH2, a male, were incredibly close maternal relatives. They both belonged to the rare maternal haplogroup R2. This strongly suggests they shared a recent maternal ancestor coming from the exact same female lineage within the village. AH4 was a different clan. This was a female, belonged to a completely different maternal haplogroup, T2C. But the T's still match in with my G's. While her broader nuclear DNA was identified to the rest of the village, she represented a different maternal line, hinting that the village was comprised of multiple distinct external maternal clans. The paternal line, the father line, the females obviously didn't carry that, but the male one did. So AH2, the adult male, was found to carry the Y DNA haplogroup J2B. This discovery was groundbreaking. It's one of the earliest recorded instances of haplogroup J in the world, a lineage that would later heavily define the genetic landscape of the Near East, Caucasus, and South Asia. So, R2 maternal haplogroup, T2C, and G1C3 are all part of the same ancient maternal family tree, tracing back to a single overarching lineage known as macrohaplogroup R. Their connection is a story of three different twigs branching off from the same deep Paleolithic branch at different points in the human prehistory. The deep origin. Every person carrying R2, T2C, or J1C3 shares a direct maternal ancestor in macrohaplogroup R. This genetic matriarch lived during the Stone Age, likely in Southwest or Central Asia. She passed down a core set of mitochondrial mutations that all three lines still carry today. So next in the Mesolithic I period is an Iranian prey of carnivores, stroke Iranian early farmer. From Iran, I match 0.14%, genetically closer to the sample than 86.28% of other genome link users.